In this video, we will continue our discussion about G protein coupled receptor inhibition reversed CD8 plus T cells effect. So, in this, the penalty in the figure shows that when this G protein coupled receptors inhibitions using this PTX is done, then this CD8 naive CD8 plus populations in the aged CD8 plus T cells alone, which has been suppressed, is again rescued, and it is similar to the young CD8 plus T cells treatment. And similarly, when they checked it about the effector memory CD8 plus T cell population, it is increased in the CD8 plus T aged CD8 plus T cells treated mice, but it is rescued in the condition where aged CD8 plus T cells are treated with this PTX. So, and it is comparable to the young CD8 plus T cells. So this shows that the G protein coupled receptors helps in reversing the impact of the aged CD8 plus T cells. Afterwards, in order to find out why these changes are happening and what are the genes that are reversed, they did the single nucleus RNA-seq analysis of these different conditions, and they are able to find out the different populations that are present in these samples. And in the penalty, they separated those populations based on which samples, based on the intensities of these genes found in different samples that are shown in this penalty. Afterwards, they see that in the condition where young CD8 plus T cells are treated with mice, and the aged CD8 plus T cells plus PTX treated with mice, they are able to find out that most of the genes are same, indicating that there is almost of the genes are been reversed. Most of the genes are been reversed. So, and when they did it, so this panel H comparisons, it shows that is the comparison between this aged CD8 plus T cells and this PTX inhibitions. So in this case, these 2, 4, 6, 7 genes are differ. So this could be the genes that are again reversed back to this young CD8 plus T cells. And when they check where these gene distributions are occurring, mainly it is mainly occurring in this excitatory neuron part, that is CA1, DG, CA2, 3 in the aged CD8 plus T cells and in the PTX inhibitions also. Afterwards, they wanted to find out what are the genes that are have this bidirectional functionality, and they can see that around 5182 genes are found to be bidirectional, whereas only 2 genes are found to be unidirectional. And out of these bidirectionals, this CA1, DG, and CA2, 3 are dominant, as these are expected because these are the ones that plays a major role in this PTX inhibitions and also in the aging T plus T cells alone. Afterwards, they use the upset plot to find out where these changes occurs, and they are able to see that most of these changes belongs to the individual cell populations, that is either in the excitatory or in the CA1. And thereafter, they did the gene ontology analysis to find out what are the pathways that are changed in the different conditions. And here also they are able to see that these young CD8 plus T cells and these PTX inhibitions pathways are almost similar, whereas aged CD8 plus T cells pathways are different. So this is true. They checked it for excitatory neuron, CA1 pyramidal neuron, and innate gyrus granule cells. And for all these three all these three systems, or all these three cell systems, they are able to see that young CD8 plus T cells and these PTX inhibitions, aged CD8 plus T cells, have the similar signaling pathways changes, whereas in the case of only aged CD8 plus T cells, they are able to see down regulations in these pathways. And this shows that in terms of the pathways also, this G protein coupled receptors has a role, and by inhibiting the G protein coupled receptors, they are able to recover or rescue the effect that is done by the aged CD8 plus T cells. This is what they have finally conclude from this figure, that the G protein coupled receptors plays an important role towards the changes that they have seen when they compare it with the young CD8 plus T cells. And these things can be rescued or reversed when the G protein coupled receptors is been inhibited, and here the inhibition is done using the PTX. That's all for this video. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.